My name is Wei. Thank you so much, Julian and the aliens bring me here to share our stories about how we model PCDH19 in our unique in vivo models. And I'm just want to say I'm truly humbled to be here today. This is my first meeting to see the families and to see the video. I will remember that video for a very long time. And this is a very much a group effort from Dr. Jack Parent's lab. So I'm really fortunate to be able to present this effort here to share the story with you. And this is the outline of my talk. You see there's many names of the PCDH19 female limited epilepsy, PCDH19 related epilepsy. So I just wanted to, I would like to have everybody on the same page. So give the introduction of the basis for our research, why we set up these models, and then why these models will help us to understand the PCDH19 function, or eventually to identify PCDH19 pathogenic mechanism for us to be able to find a cure. So the second talk will be presenting the very preliminary data we got from the iPS-derived neuron cultures, and then lastly, I will present the progress developing a novel virtual FLE. Later on, we'll call PCDH19 female limited epilepsy FLE patient models for us to further identify where PCDH19 expressing in the human cortical developmental processes, and hopefully understand more and better about PCDH19 function, and eventually to shine light on the pathogenic mechanisms of PCDH19 FLE. I think everybody in this room see these figures, sort of similar figures before. So the PCDH19 FLE is very unique X-linked inherent orders, affecting females, girls primarily, and homozygous males were not largely unaffected and asymptomatic due to these two extra chromosomes activation and inactivation state. So far, there's many mutations has been found. These are just selected variants, and has been identified PCDH19 gene. You can see many of them clustered in exon 1. Exon 1 is a really important domain for PCDH19 functions. Intermediate, the extracellular cat-Henry interaction is a extracellular cat-Henry interaction domain is really important for PCDH19 functions. That these mutations clustered in this exon 1 encoding for this EC domain speaks a lot about PCDH19's function immediate cell-to-cell adhesion. As I alluded before, PCDH19, it is subject to X chromosome, subject to random X inactivation. In the stem cell stage, in the single cell, you have two active chromosome. One is wild type, but the other one is mutant, shown in green and red. And then during early embryogenesis, one of the chromosomes was subject to inactivation. This is selection process is random. And when the wild type has been activated and the mutant has been inactivated, you have one cell type has PCDH19 wild type cells. And then, in contrary, if the wild type chromosome has been is activated, then there will be PCDH19 mutant cells containing PCDH19 mutant proteins. So the mechanism of PCDH19 FLE has been best explained by this called cellular interference model. This model not just for PCDH19, a few other X-linked genes like craniofacial nasal syndrome also has similar patterns and a similar mechanism as PCDH19 FLE. The gene caused the CFNS syndrome is the AMPHA gene. So when this individual has only one type of PCDH19 protein, the individual will be healthy. And when the carrier males has mutant PCDH19 protein, and then also seem to be healthy in general. The problem comes is when two types of cell type mixing together, so the cell cannot be interacting well. As illustrated here, maybe the mutant cells are segregated by the wild type cells, and then this is the basis for the standing hypothesis about how PCDH19 causing female girl-specific epilepsy. So this abnormal segregation in PCDH19 heterozygous brain might result in altered network brain activity. As for how, we have known little about it. So I really should see intrigued and impressed by the work has been done by Dr. Paul Thomas, and this heterozygous mouse of PCDH19 nicely illustrated the cell segregation in the PCDH19 heterozygous mouse, and leading to a segregation of two populations of the cells, and therefore observe the increased network brain activity by his electrocorticograph analysis. However, as there is no seizure phenotype has been observed even in the heterozygous mouse yet. So what we think is it might due to the developmental differences between the mouse and the human. That being said, the mouse is a very good model and an exciting model to study PCDH19. And so the differences between the human and mouse neurogenesis is human has this the expanded SVZ area, particularly the subventricular zone, containing a large number of the radioglia cells, which continue to proliferate even in the adult human brain. So this feature could contribute to the different sizes of between mouse and human, and also could explain why the human has more folding surface and have more capacities for the neuron density. Therefore, our lab, Jack Parent and Dr. Jack Parent are very interested in using a human-based model to model PCDH19, and then we can study PCDH19 FLE in human genetic background. So we will look at PCDH19 function using this in vitro model to model the human cortical development and neural network in vitro, and looking for early cortical development PCDH19 in the early cortical developmental processes. As we know, PCDH19, some of, I speak to Julie, like up to maybe 30–50% of the PCDH19 affected individual girls might have autistic features that indicate that PCDH19 play an essential or important role in the early and developmental of the cortical. And also looking for the neuronal network function too. So using the mutant model, we could compare to the normal function, identify some of the pathways and the molecular mechanisms of the PCDH19 FLE, which could help to identify target-based therapeutic method. So this is more like a perfect cycle of life. So you have, we think IPS has a dramatic promising future for modeling the disease, also correcting the mutations, therefore to correct the mutation, and then hopefully can correcting the phenotype or the symptoms of the patient. So our lab taking the approach is reprogramming the skin tissue or blood cells back to the potency of mimic the stem cell state, can differentiate them into neurons. And the meantime, if we can correct the mutations in the health in the IPS stage of the derived from the patient, then we can compare the morphology of the corrected mutations with the mutant cells, and and then we'll be able to give insight into some of the mechanisms and also help this model system also easier to perform drug selections and then screenings, and therefore help to future drug screenings. First, we did is thankful the generosity of the families from the foundation, so we'll be able to obtaining a few of the patients' skin fibroblast, and when this work done by CC Du is a talented MD and PhD student in the lab, and then she using the reprogramming method, reprogram two different type of patient. One has a missense mutation, and in the critical domain exon 1, and the second one is the 15 base pair deletion mutation patient content mutation. So the panel B is the showing these reprogrammed pluripotent stem cells has a proper expression of the pluripotency genes, which is important for these cells to be differentiated into the cell types we would like to use to model PCDH19 FLE later on. And then we have done chromosome karyotyping to make sure the karyotype is normal, and we also did in situ and Sanger sequencing to make sure we are really see the mosaic expression of two different alleles in this population. We don't want to see like in panel D, there's no cyst. Cyst is the inactivation mark for X chromosome. So if cyst is gone, that means two chromosome has been activated, which is not good model for model of PCDH19 FLE. And then we also have done the RT-qPCR to check PCDH19 levels in the cells we derived. And think about the chromosome, X chromosome inactivation. So you should really see the reduced level of the PCDH19 compared to the other wild type control stem cells as showing here. So these are three different lines, and this is the control cells, and this is two different primer set to targeting the beginning and the end of the PCDH19 transcript. So all these three lines showing a great reduction of the PCDH19, but still maintaining PCDH19 expression. That means part of the population is PCDH19 wild type, and part is mutant, a loss function of the PCDH19. As I alluded in the previous slides, using the iPS models derived from the patient is a great model. We can look into the patient genetic background directly, but however, we needed to monitor the X activation state constantly to make sure we are modeling this mosaic expression of PCDH19 in every stage of our experiment. So the complementary approach we took in the lab is using CRISPR Cas9 technology, as already introduced by Dr. Paul Thomas in the previous talk. So in using Cas9 CRISPR gene editing system, we could introduce loss function alleles into control male cells. That way we can mix the male wild type cells with the male knockout cells by the CRISPR, and then generating this mixed population of the wild type and the mutant PCDH19 cells. So these will be more consistent expression levels in this mixing culture, as also done by the first speaker, Isabel, in the talk. So just brief recap on the Cas9 gene editing approach. So Cas9 is a nucleases. We're cutting the nucleotide randomly if there's no guided sequence to tell the Cas9 where to go. So that's why there's a guide RNA sequence guiding the Cas9 to the PCDH19 locus, specifically cutting the PCDH19 alleles. That being said, there will be off-target effect, but there are ways to monitor that. So we have been using a male control cells and then using the guide RNA, introduce indels, therefore trigger the loss function of the PCDH19 and this locus. So the powerful of this approach is we have isogenic control where we generate the mutant. We're looking at the same genetic background as the mutant and the control cells. And then after we obtaining these cells, functional study, we are focused on generating cortical-like excitatory neurons. And then this is a method modified from 2012. It takes about 4 to 8 weeks can generating functional, still immature though, cortical-like excited neurons. It's starting with plating the IPS we generated previously, and then using inhibitors called dual SMAD to force the dorsal expression of the neuronal markers. Therefore, we can generating the cortical-like neurons. So this is the IPS. The green is the OCT3 the nuclear protein for pluripotency, and the SSEA4 is the memory memory markers for the pluripotency. And then at the next stage is this monolayer IPS will become a really structured epithelial sheet. In the center will be the apical center, mimicking the SVZ area we talk about, I'm showing in the few slides before. And then there's a large cell population is PAC6 labeling the neural stem cells, essentially giving rise to all the neuronal types in the brain development. And nesting labeling also the neural stem cells will later on being induced or derived into different type of neural cells. Now, this is a typical imaging view of the cortical-like excitatory neurons, and the MAP2AB labels in general mature neurons, and VGLU1 labeling the mature cortical excitatory neurons. Anchor-G is the another marker to label that. So first question we ask is, based on the in vivo data and the human in situ hybridization data, PCDH19 expresses throughout the cortical development stages. And then in our in vitro system, can we identify where PCDH19 is and whether the expression of the PCDH19 pattern really matches what we learned so far. So we're taking samples from these 3 different stages, IPS, neuron rosette, and also cortical-like excitatory neurons. And then we did a RT-PCR to quantify the mRNA level of the PCDH19. And as you can see, these are the 2 different locations of the mRNA of the PCDH19. PCDH19 is expressing in both neuron and neuron rosette, but expressed highly in the neuron rosette. That indicative PCDH19 does has a very important role to regulate in the early human cortical development. So the next experiment CC did is since we see this high expression level of the PCDH19, so in the patient-derived neurons, whether the structure or morphology of the PCDH19 has been disrupted or has been altered. So the answer is indeed it is. So she has done stainings for the apical center, the lumen the junction of the neural rosette by N-catecholamine antibody. I think she has a hard time to find an antibody works really well for the immunostaining or even other biochemical works for PCDH19. So at this stage, she didn't have the PCDH19 staining to show PCDH19 also being disrupted or mislocalized in this apical center. So this is the control IPS, and it's a healthy individual. To be noted is a healthy state. However, this is not isogenic control of the FLE patient IPS, and it takes quite a lot of effort to generate the correction mutation and getting isogenic control. I think we are working on that, but now this is just a unrelated healthy control. And then in the FLE, the neurons from the FLE patient, you expect there's two populations of wild type and the mutant. So she observed a disrupted uncatechering localization pattern in this patient. And as I said, we have this male patient line generated by CRISPR. So we also mix the isogenic male patient isogenic CRISPR line with the control line together to see whether we can see the same phenotype. The answer is also it is. So this is the male control male knockout line only, so coded as healthy state because we expect this is a female specific. And then when we mix the male control line, this male control line with the male CRISPR knockout line, also the uncatechering localization patterns is quite different from the wild type. And then next, we look at the activities, the neuronal activities in the excitatory neurons we generated from this experiment. And first, she did a morphological analysis on these cortical-like neurons. So the top panel shows the healthy IPS line-generated neurons, and the bottom is the female patient-derived, the neuron derived from a female patient. And compared to the healthy control, the female patient, the neuron generated from the female patient tend to clumpy, aggregate, and it shows this in this left panel and also in the middle. And then they have more self-crossing, and there's publication showing PCDH19 is important to prevent cell crossing. And then there's a more neural outgrowth per soma. So if you look at this neuron in here, so this is the soma, and there's many processes growing out from the soma, and compared to this, the wild type has really thick and then less neural outgrowth from the soma, which also, when we see this data, we suspect this reflecting a premature neuronal differentiation. So therefore, we stain the culture with the different antibodies recognize different layers of the neurons in vivo. So the TBR1 is the markers for SVZ, where the neural progenitors proliferating dramatically. So you can see in the control cells, there's a lot more TBR1 positive. That means there's a lot of neural progenitor cells in the control cells. And in the patient, there's dramatic reduce of the TBR1 staining. And in complementary to this TBR1 staining, the CTP1 is the upper layer neuronal cell marker, and the SAB2 is the deeper layer neuronal cell marker. So as compared to the wild type, the female has a lot more SAB2 and the reduced level of the CTP2 that indicate the female, the neurons generated from the female patient has accelerated neuronal maturation. And then we confirm this result by using the mixed culture of the male controlled mutant neurons generated by the CRISPR. So, in this experiment, we label two different populations with two different colors. And then for the control male lines, we label with GFP lentivirus. And then for the mutant line, we with RFP lentivirus. So the cell can be stable expressing the GFP and RFP at a time when mix them at IPS stages. So when we look at the patterns of the distribution of the GFP versus the M-cherry neurons in the wild-type cells, the healthy cells. You could see the M-cherry and the GFP cells growing, mixing together quite nicely. However, in this female, this FLE state, the GFP cells is the control cells, doesn't really like to mix in with the M-cherry, and they always tend to be stand by itself. I think this experiment is still very preliminary, so we need to repeat this, that indicative the segregation between two different populations, and then zoom in morphology of these FLE neurons, you could see there's more outgrowth compared to the healthy controls, which is another validation of the studies we get from the neurons generated from the patient iPSC I showed in the previous slides. And then furthermore, we want to see whether this, what's the activity of the neurons from this patient, whether they have activity as we observed before in the in vivo model, and what's the activity like. So the assay we took is called multi-electro array recording. And this platform is from axon by system biology. Is quite powerful. We can monitor the neuronal activity for months, because the neuron was grown in this each one of these wells, and each wells has the electrode to recording the activity of the neurons, and then these neurons growing on there can be grown for months, so we can do a longitudinal tracking of the neural activities for the patient and the control, and then in future, this could be very useful for drug screening as well, because this can be done in the 96 well or 12 well. So potentially you can screening drugs looking outcome using the activity as an outcome, whether it's reduce or increase the activity. I think our experiment purpose is try to identify a phenotype, and then we can use this platform to for drug screening. This is the terminologies I'm going to show in the next slides. I don't think I can understand it fully so far now, but just pointing to what we're looking at. So the this is the plot from the recording. So the white spikes is the spontaneous activities, and then the red one is the bursting activities. We can measure how often this bursting activities, also how long the bursting activities by different formulas. And then the bottom shows the network bursting duration, so how often this neural firing. So it's very exciting to see the neurons generating from the male patient IPS actually has all increased firing activity as measured by the mean fire rate, also the number of the burst, and also the burst frequencies increased compared to the control neurons from the control iPSCs shown in this blue. So at this point, we are trying to finishing this study and then do more comprehensive validations what we got so far. And but we are confident what we saw here is very exciting findings, because these are done with different lines and then 4 to 5 different replicates. That being said, I want to summarize what I have to explain so far. So we have derived multiple IPS lines for multiple patient families, and these lines are really valuable sources to model PCDH19 female limited epilepsy in human genetic background. And then using these lines, we'll be able to identify FLE-related phenotypes, which is really important for us to moving forward, and to identify I think in the long run, the drug target of screening. And as I showed before, this neural rosette derived from the patient lines has altered lumen structure, is the SVZ center of the neural rosette. And also the neurons has migration and morphological abnormalities in the patient neurons. And most excitingly, we see increased excitability from the patient neurons. We feel confident these lines will give us essential tools and providing very important clues to determining how this cell-to-cell interaction and cell aggregation between the wild-type cells and PCDH19 cells could lead to the excitability, also seizures. However, one question have been really bothering us about. We wanted to know since I alluded before, we had a hard time to find a PCDH19 antibody to really pinpoint whether PCDH19 is in the center of all this phenotype. Therefore, we wanted to moving this research to a additional level, developing tools for us to be able to tracking PCDH19 expressions while tracking the phenotype. As you can see, PCDH19 is why this is challenging. PCDH19 is belong to these large protoheric families, and they are have conserved domains and either extracellular or cytoplasmic. Finding a antibody and validated antibody is quite challenging. The approach we took is similar to what Dr. Thomas has been presented in his talk. Actually, in the very beginning of setting up this system, I have really useful discussions from his talented postdoc, Daniel, and to how to set up this in human stem cells. Again, go back to this Cas9 system. One part of the Cas9 system is introducing indels. You're cutting the genome and then introduce unwanted indels, therefore degrading the mRNA and the protein. The second mechanism of the Cas9 mechanism is if we tell the Cas9 nucleus cutting PCDH19 particular location, if we tell the cell, give the cell a repair template, and the repair template can contain any of the sequence you wanted to put in, then the cell will use its native repair mechanism, will integrate this sequence of your interest into the genome. Further illustrating is this is how we design our in-frame tagging of the PCDH19. In the last coding exon, so the black is the stop codon. We try to put in a HA flag tag, and before the stop codon, and also in frame, we won't introduce any of the artifact overexpression, and also the C terminus. The last codon is intercellular, so we can tracking PCDH19's expression pattern. As I said before, this is really precise gene editing, so we really just putting this tag into the PCDH19, not other PCDH proteins, and HA and flag has really high quality antibody. We can specifically identify where PCDH19 is, and another advantage of this system is we could use this one antibody for all purpose experiment to reduce experimental noise, like biochemical studies, cellular studies, and then molecular studies. So I have generated a few lines using this method, and one of the lines is a homozygous tagging of the PCDH19, and the second line is heterozygous tagging. One allele has been tagged, and this is the H9 embryonic stem cell non-tagged lines. So we wanted to confirm this manipulation of the PCDH19 won't interfere PCDH19's function also won't change the pluripotency of the stem cell after editing. So we can use this cell line to differentiate into neurons and then other applications. So as shown in this also RT-PCR, and the PCDH19 level in the tagged lines, the first two really comparable to the untagged, the PCDH19 level in the untagged lines. And then the pluripotency marker show the pluripotency in these two lines also maintaining a very proper level. And that, again, SSEA4 is cell surface marker, and this is the nuclear protein labeled the pluripotency. This is fairly a new progress. We haven't done exhaustive expression or a localization study yet. So I'm going to show a couple of slides we have done using this system to show the PCDH19 localizations. Primarily, we focus on the neural rosette stage, where we observed there is a high expression levels of PCDH19 in our RT-qPCR experiment. So the top one is the tagged PCDH19 embryonic stem cells. So the bottom is the control, the non-tagged PCDH19 embryonic stem cells. So we use the similar differentiation protocol as we showed in the patient neurons, and taking the sample from the neural rosette using two different antibody, because we put in two sequence HN flag. They are nicely co-localized into the adhesion junction in the neural rosette, and then this is quite specific, very specific to only the tagged lines. And as from our experiment from the patient-derived neural rosette, we see N-cadherin localization pattern has been altered in the patient lines. So the simple experiment I did is co-localizing HA to identify PCDH19 and with N-cadherin to see whether they can co-localize what's the relationship between the PCDH19 and N-cadherin. And this is the nuclear counter staining, and this is the merge between the HA and N-cadherin. It's nicely co-localized with N-cadherin in the adhesion junction. Again, which is a reminiscent structure of the SVZ of the human cortical region. In addition to this 2D neural rosette experiment, our lab has started to generating mini brains in a dish. We also call that is a human cerebellum organoids, is really a 3D assay of the human brain development. And we have been able to generate these organoids from different protocols. Here I just show you that one of the protocols we're using published 2006 by Qian et al. So starting with the iPS cells, and then similar to the 2D, we adding the dual SMAD and then induce the ventral, the dorsalization, and then and then we can grow this for days and 100 days, and then later on adding the nuclear factors to increase the maturation of the organoid. This is the proximate size of this organoid, and this system can really recapture the brain layering structures in the cortical regions. And most strikingly is we could be able to generate the SVZ, the out there expanded SVZ regions in this system. So this is how we call span omega. So the organoids was in these 12 wells, and then using this motorized system can mixing the cultures, increasing the oxygen level in the wells, and then when we're taking day 60 organoid section them to staining for markers for different layers, we could see the nice structure of the neural rosette, the SVZ or VZ like areas in the middle, and then the CTP2 and the SAB2 for the deeper upper layer markers. As I mentioned before, this system could be also very useful to generating the SV, the out layer SVZ area, which containing the out radioglial cells, important for uniqueness of the human cortical development. And then what's PCDH19 look like in this 3D organoid? So the panel on the right is the HA staining, shown in the green, and then this is the TUG1 staining showing the neurons generated in this organoid. And then we overlay these two, we can see apparently PCDH19 still localized to this cortical, to this adhesion junctions, the SVZ center of the apical center of the SVZ area, and it might not be clear. The HA does not quite overlap with the TUG1 signal, and based on our preliminary observation, I think it seems in between the neurons that really the cell-to-cell adhesion surfaces. So the one on the right is the older organoid from this experiment is day 76. You can see the neuron rosette grown bigger, but still a PCDH19 in the junction center and starting to also labeling more of the surface or junctions between cells. So as for what cell type, we haven't gone to look for a complete panel of co-localization with markers identify other cell types, but our future studies wanted to do this in a very detailed way to co-localize this HA PCDH19 with different cell markers marking for different layers of the cortical and a different cell type of the cortical. But we are very excited. This very new, unique. So as I said that, so our future direction is try to knock out PCDH19 in this tag line and then mixing these two cell types together. So we could be monitoring the PCDH19 expression while also generating this mosaic expression of PCDH19, so we can directly pinpoint where PCDH19 is, whether PCDH19 causing this phenotype directly. And with that, I think I already summarized the whole talk previously. So we think combining the CRISPR gene editing and the stem cell technology, we'll be able to develop this powerful in vitro platform to model PCDH19 in vitro in human genetic background, can help us to understanding PCDH19 function during human cortical development, therefore identify specific related mechanism of PCDH19 FLE. Lastly, I would like to thank Jack Parent. He's a tremendous mentor and also very brilliant in helping designing the experiment. And again, CC is the graduate student put a lot of work on developing the IPS derivation from the patient cells, and did all those differentiations and showing this nice FLE-related phenotype in the IPS neurons. And Andrew has been helping with gene editing, and Yuan is another physician and clinician in the lab, also involved in PCDH19 work. Not lastly, last not least, but I think all this work cannot be done if we don't have this family support and generosity to donating the tissues and then the informations. And then the work was funded by NIH and Q Syndrome and also PCDH19 foundations from Italian group. And thank you. I will take questions.
Any questions? Yeah, Joseph.
Yeah, that's a lot of interesting work there. I was going to ask you the IPS lines you generated from females. Have you separated the two cell types, which the wild type and the mutant chromosome active? And if you haven't, how do you know that the cell lines over time don't skew towards one X chromosome expressing cell type? Do you monitor it in some way?
So, this is always outstanding questions when we're doing the experiment using the female lines. And we have been using primarily 3 different approaches to try to address, validate the state of the chromosome. One is the Sanger sequencing of the cDNA. We want to see 2 expressing alleles. And then the in situ by the cyst. And we haven't gone to work with the PCDH19 in situ probe yet. And then also we do the RT-qPCR to make sure we do observe like 50% around. But we haven't gone so far to do the estrogen receptor analysis yet. Those seems to be another good approach to monitor this mosaic state. But that being said, we feel it's quite a lot to tracking this. That's why we wanted to go back to the basics, making this mixing expression cultures with the antibody can recognize PCDH19 in vitro and in real time.